Students, this is a podcast that contains information regarding exceptions to Mendel. And what you've learned in the past is that typically in a monohybrid cross, when you have a heterozygous and a homozygous dominant, that the phenotype is a dominant trait. But there are some exceptions to that rule, and we want to go over those exceptions. So there's 3 documents that were passed out. The document which you see on the screen here, multiple alleles, which covers blood type, and sex determination and sex-linked traits. So as you go through this podcast, it's just one big long podcast. Go ahead and pause it to do the problems, but your goal is to complete all 3 documents and turn them in by the end of class. Okay? And you will not get credit for them unless you watch the podcast and then you've answered all the questions. So the first 2 exceptions to Mendel's rule deal with incomplete dominance and codominance. Just want to scroll up just a little bit. There we go. So incomplete dominance is really about the idea that there really is no dominant allele. And so typically when you have 3 combinations, you have, let's say, the homozygous dominant, the homozygous recessive, and then a heterozygous version. Typically, we would say that this genotype and this genotype would give you the dominant trait. But that's really not what we see in all cases. So there are some exceptions. And incomplete dominance means that there's a blending of the traits. It's kind of like mixing paint. If I mix red paint and white paint together, I get pink. And oftentimes in flowers, we see that mix. So here's an example in snapdragon flowers. Snapdragon flowers show this pattern where there's a red allele right here, and there's a white allele over here. I'm just going to erase this to the side. Just give me a second. And the genotypes are over here. So when you have the homozygous red, we'll represent those as RR. When you have the white version, it's the homozygous WW. And then here we have the heterozygous version where you get a mixture. It's literally just like mixing paint. Red and white make pink. So the example is cross a red snapdragon with a white snapdragon. So a red snapdragon is, of course, big R, big R. The white snapdragon is, of course, WW. And what you get in the Punnett square are all individuals that are pink. So if you look at the ratios, and in this instance, the ratios are all going to match up. They will all be equal to each other. So the genotypic ratio are 4 RW and 0 everything else. That also gives you 4 pink. 0 everything else. So typically in monohybrid crosses, you have 2 phenotypes, the dominant and recessive. But in this case, you actually have 3 phenotypes. You have the 2 colors separately, and then you have the mixed version of it. So incomplete dominance, there's no dominant or recessive alleles. There's a blending of the traits for the heterozygous version. All right, it's like mixing paint. Okay. So now you've been given some information about what incomplete dominance means, and you can complete the questions. So I'm going to erase this and then go down to the next section, which is about codominance. The computer froze just a little bit. There we go. So codominance now. Codominance. So codominance is when both alleles may be present in the heterozygous version. So if incomplete dominance is mixing paint, codominance is like mixing Play-Doh or clay. We did that in the cell cycle lab. And I asked you, please do not mix the colors because you can't unmix them. All right? Codominance is when the heterozygous version gives you a little bit of each version. So in this case, we see this in birds, we see this in chickens. We have black chickens, we have white chickens, and when we have the heterozygous version, you actually get to see both black and white feathers. Right? We have that mixture just like Play-Doh. Right? When I mix red and green Play-Doh together, I don't get a separate color. I get a big ball of just red and green Play-Doh. All right, so here's our example. Cross a black and white chicken. So I got my black chicken, I have my white chicken, and all of the offspring have both black and white feathers. So the genotypic ratio again would be 4 BWs to 0 everything else. And then you're going to see 4 black and white chickens to 0 everything else. And again, in that phenotype here, because it's an exception, we now have 3 phenotypes as opposed to 2. So just a little bit different. One way to always check yourself is that the genotypic ratios and phenotypic ratios are always going to be the same. Always. So if they're not, then you've done something wrong. But now that you've been given an explanation for what these 2 exceptions are, go ahead and pause this and answer the rest of the questions on the worksheet. And when you're finished, go ahead and click play again, and I'll go over the multiple alleles section. So hopefully you had an easier time completing the exceptions questions. You should have completed questions 1 all the way to 6. So here we have what are called multiple alleles, another exception. And multiple alleles means that the traits are controlled by more than 2 alleles. Now, typically, again, you have 1 dominant and 1 recessive, or if you had a mixture of the 2, then you've got the codominance or incomplete dominance. But here, we have 2 alleles that are going to be dominant to another, and that's what blood types is a really good example of that. So in blood types and blood grouping, I'm going to kind of scroll in here. Just give me a second. Here we go. In blood grouping, we have multiple different types of blood types. We have A blood, B blood, AB blood, and O blood type. If you're not quite sure what you are, then either ask your doctor or if you've ever given blood before, which is how I found out, you get a little card from the Red Cross giving you your blood type. But really all we're looking at are proteins on your cell membranes. Okay, we talked a little bit about this a couple units ago about those cell surface markers, and if you have A blood, you've got the A protein, B blood, you have B protein, AB blood, you have both, and O, you don't have it. So it's just a way to identify what type of red blood cells that you have. So these are the blood groups, and what it really means gene-wise, and what it these are the phenotypes, of course, the blood type that you have, but the genotypes are a little different. So we're going to use the capital I. We're going to use capital I as our dominant gene, and lowercase I is our recessive. And we're just going to symbolize what type of I we have. Is it going to be the A allele or the B allele? So in A blood type, if you are homozygous, that means you are IA. That means you're homozygous A, right? Where you can only donate the A gene. But you can also be heterozygous. You can have IA, lowercase I. Right? Either way, that makes you A blood. B blood is very similar, but just the opposite. Right? Only we're using B now instead of the A. So I have IB, that's a homozygous individual, or you have heterozygous B. Either way, you have B blood. AB blood is a little different. AB blood just means that you have one of each. You have the A allele, and you have the B allele. And lastly, for blood type O, since that is the recessive gene, there's only one way to get type O blood. You're going to be O. So if we want to look at being able to donate blood or be able to receive blood, we're going to kind of add on to this over here just so you understand what it is that we're talking about. That's why I kind of left this space here. So I want to say right here, donates and receives. I'll draw my line here. Well, kind of a bad little box there, but no deal. I'm going to switch to green here. So if you were an A blood, all right, think about who you can donate to. You can donate to the A people because they have A blood. You can donate to the AB people because they have the A gene. And that's about it. Nobody else. If you donate to B people, that means that their body won't recognize the A gene and they will reject the blood. If you donate to O people, they won't recognize it because they don't have it. So you can only donate to A people and AB people. A individuals can also receive blood from A people. You can't, although you can donate to AB, you cannot receive AB because your body won't recognize the B protein. You can also receive blood from the O individuals. Your body, for some reason, is able to recognize or not fight against typically those O people. They don't have that protein on the surface. So it's not seen as an invader. If you're B blood, you can donate to B people and AB people. And that's it. You can receive blood from B people, not AB people. Again, although you can donate to the AB individuals because they have the B protein, you cannot receive blood from the AB individuals because your body doesn't recognize the A protein. So the only other blood type you can receive from is type O people. AB people are a little tricky, right? Some good, some bad. If you're AB, you can certainly donate to AB people. And I'm going to erase that, and that's about it. Whoops. See if that happens. Nope, it didn't erase it. My computer's just a little slow. So you can donate to AB people, and that's it. If you donate to A people, that B gene gets in the way. If you donate to B people, the A gene gets in the way. So you can only donate to AB people, right? But you can receive blood from A people, B people, AB people, and O people. So therefore, if you have type A blood, AB blood, which is the rarest of the kind, you are known as the universal receiver because you can typically receive blood from anybody. O people, on the other hand, are just a little different, right? Just a little different. So O people, who can you donate to? Well, you can donate to A blood, B blood, AB blood, and of course, O blood. So you are known as the universal donor because you can donate to anybody, but you can only receive blood from O individuals because if you're given any of the other types, your body won't recognize those proteins and it'll be rejected. All right. So again, AB individuals are known as the universal receiver. O people are known as the universal donors. Okay. The other little bit of like stats, if you look at world health statistics, about 40% of the people on this planet have O blood. And you might think, well, why is that important? And I'll tell you why. Hospitals want O blood because if you, again, look at the chart here, O people can donate to anybody. So in any given situation that's an emergency, of course, hospitals will try and match the blood. Of course, that's the number one priority. But, O people, there's number one, 40% of the people are O blood. So you want to have that the most anyway. But, in an emergency situation, when you're not able to find the blood type, of course, you can go ahead and just hopefully give that individual a blood transfusion and hope that it kind of works because typically, again, anyone can accept O blood. So it's really important to really stockpile the O blood type. All right. So that's a little bit about the history of blood types. Let's go ahead and do a couple of problems. I'm going to erase this. Did it work? No, it did not. So I'm just going to have to erase it the old-fashioned way, which is fine. And we'll do a practice problem or two, and then you'll be on your own. So I'm scrolling down here. So now it's your turn. So a father is O, a mother is O. Let's see what that looks like. So the father is O, so they're little I, little I. Mother is O, little I, little I. What do they have? All individuals with O blood. So what does that mean? 100% O, 0 everything else. And that's a problem. Father is type A, homozygous. So go back to your chart, and a homozygous A individual is IA, IA. Both of their alleles are A. The mother is type B, homozygous. Remember, there's two ways to be type A, either homozygous or heterozygous, and two ways to be type B, homozygous or heterozygous. So a type B individual, IB, IB, and then in the Punnett square, what you have, AB, 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 and AB. So mother is A, father is B. What you get is 100% chance of an individual being type AB blood. Right? So that's very helpful when determining paternity cases to doing simple blood tests. And it's also important when we're looking at some of the blood diseases. But go ahead and pause this podcast and complete the rest of the questions. There should be 3 all the way to number 12. All right, make sure you're showing your work. When you're finished, hit play again. Okay, hopefully you had a easy time working with multiple alleles. My suggestion to you is if there were any ones that you were not sure about, go ahead and circle them. Right? Do not just take the easy way out and copy off your neighbor. That does you no good. In fact, that'll ensure the fact that you fail on the test. It's better just to circle it and ask me a question, even if you write it on the paper. Okay? It's easier for me to fix that way, but if you're just going to be lazy and copy and cheat, that's not going to get you anywhere. So the last type of problems that we want to do are called sex determination or sex-linked traits. So let's back up a bit and just review that. Again, the first, there are 23 pairs of chromosomes in humans, 46 total, 23 pairs. The first 22 pairs are called autosomes, meaning they do not determine your gender. That 23rd pair does determine your gender. Those are called sex chromosomes. And we know from previous studies that males are XY, meaning that the sperm are either going to carry an X chromosome or they're going to carry a Y chromosome, and that females are XX, meaning that it doesn't matter what type of egg they produce, they're always going to contain an X chromosome. So again, here's a little example of the Punnett square. And I show this to you because every single Punnett square you do with sex determination or sex-linked traits starts with this. You can diagram exactly this picture, and all you have to do is label the X's in your problem. So again, father XY, mother XX, and when they go through meiosis, the dad is either going to make X sperm or Y sperm. Now if they don't, and you somehow get an XY on one side and nothing on the other, that's called non-disjunction, and that can give you something like Turner syndrome or Klinefelter syndrome, something we talked about with the karyotypes, right? Mothers, it doesn't matter. When they separate in meiosis, you're always going to get X eggs. So over here, again, this shows the gametes. If this sperm and this egg meet together, it could be a girl. If that sperm and this egg meet together, girl. This sperm and that egg, boy. This sperm, that egg, boy. So on any given occasion, it's about a 50-50 chance that you're going to have a boy. Or it's a 50-50 chance you're going to be a girl. Okay? So every Punnett square we do has to be exactly this picture. All we are doing now is labeling the X's, really, and I'll get to that in a minute. Okay? And again, if you look at who determines the gender of a child, it's always the male, because it depends on which sperm fertilizes the egg. If it's the X sperm, the offspring is a girl. If it's the Y sperm, offspring is a boy. So I'm going to erase this and kind of move on. But again, keep in mind that this is the premise of everything that we're going to be doing in this little lesson. So if we just review some of our terminology here. Okay. Here we go. I'm going to erase these green marks here. Okay, what are the chances of having a, oops, there we go. What are the chances of having a boy for each birth? Again, just look at your Punnett square. You should see that it's about a 50% chance. 50%. What are the chances of having a girl? 50%. Out of 4 children, how many would you expect to be female? Well, if it's a 50% chance and there's 4, that's 2 out of 4. Same thing with men. If there are 10 children, half of 10 is 5. So 5 females would be girls and 5 would be boys. And of course, we know that's not exactly what we see in nature, but if you really do look at boys and girls globally, it's a pretty even split. It's kind of odd like that. All right, so what sex of chromosomes do both males and females have? They both have an X. What sex chromosome do only males have? They have a Y. So where do males get their X chromosome from? Got to be the mom, all right? They get their Y chromosome from the father, all right? Where do females get their X chromosomes from? Both parents. All right, and why does the father determine the sex of the gender? I'm going to let you answer that by yourself, and I will check that. And again, if you look at number 12, just to review, males produce 2 kinds of sperm, the X and the Y, where females only produce 1. So now that we've done a little bit of review, let's go ahead and do some problems. I'm going to erase this and move on to the next page. And again, the expectation is that you complete these problems and turn them in. Okay, so sex-linked traits. These are traits or diseases that are linked to the X chromosome. And again, because males get their X chromosome from their mothers, and they only have 1 Y chromosome, or 1 X chromosome, males are going to be affected a little different than females. Males either have it or they don't, because they only have one X chromosome. Females, because they have 2, again, they can have it, they cannot have it, or they can be a carrier, which is a heterozygous version. In males, there are no heterozygous versions because you only have 1 X. Right? So 3 common ones are color blindness, hemophilia, and male baldness. And again, we do see on a rare occasion baldness happen in females, but typically it happens only in males. And again, this is why I blame my mother for me being bald, because I got my X chromosome from my mom, which means that that trait came from her side of the family. So we're just going to symbolize these X's with N's. It's just going to be easier that way, okay? N, capital N for normal, lowercase n for the trait. So we'll do the first several together. All right, so the genotypes. For a normal color vision female, so it's a female, so we know that it's an XX. So if there are normal color vision, we symbolize those X's with big N's. They're homozygous, right? But females, again, they can be carriers. So we want to symbolize the X's with as a female, but if they're a carrier, they're still normal, but they're heterozygous, meaning that they can pass that little n on to their offspring. Colorblind females, there's only one way to be colorblind as a female, and it's less likely because there's multiple choices. You have to be homozygous recessive. Okay? So normal, so if we go to males, males either have it or they don't. Because males only have 1, there's an XY. Right? So a normal color vision male is X big N Y. That's it. There's no other option. There's no heterozygous version. There's nothing attached to that Y. So for the colorblind male, it's an XY because it's a male, and that's an X little n Y. Right? Men either have it right here or they don't. Simply 1X Either have it or they don't. So let's look at baldness. So what is a bald male? I'll give you, it's male, so it's XY, and if they're bald, they're going to be little n, X little n y. A normal male would be XY being a male, and big N for the X because they're normal. Let's look at hemophilia, okay? A hemophiliac is when your blood doesn't clot, right? Hemophiliacs, it's really dangerous because you could, when you bruise, that is called internal bleeding. And if you're an individual that can't stop that internal bleeding, that's a problem, right? Hemophiliacs can like bump themselves up against a desk and bleed to death inside. They have no way of stopping that blood. So they have to take lots of medication to kind of get their blood to clot. It's a really horrific disease. So hemophiliac female, it's a female, so we know that they're XX, and they are hemophiliac, so that means they're homozygous recessive, X little n, X little n. A normal female, XX, non-hemophiliac, so they are X big N, X big N. If you're a carrier, you would be X big N, X little n. That would be a carrier version, but I didn't ask you to do that. I just said non-hemophiliac female, okay, normal. So a male, again, you either have it or you don't. Male meaning that they're XY. I'll put XYs right here. So a hemophiliac male would be X little ny, and a normal male would be X big NY. Right, again, there's only 2 versions for the male, 3 versions for the female, because of that X. So I'm going to go ahead and erase this, and hopefully these genotypes made sense when setting up our problems. Right, we'll do a couple of questions together, and then we're simply going to let you all try and do some of these on your own. Okay, I'm going to scroll down here to number 1. We'll do the first 2 together. A homozygous normal female marries a colorblind male, right? Homozygous normal female, so XX, that's a female. Homozygous normal means they're X big N, X big N. Marries a colorblind male. It's a male, so XY for male, and if they're colorblind, they're X little ny, right? So over here, and what you should really do first, I recommend everyone doing this, is just simply do the male-female Punnett square. What I mean by that, oops, I'm going to erase that to get my colors all the same. So I have XX, XY, and I know every Punnett square that I have is going to look like this. Half of them are girls, half of them are boys. Now it's a matter of just where do the ends go, okay? So let's start with my female. X big N, X big N. So I would have a big N here and a big N here. Big N here, big N here. There we go. Now my N's taken care of. Now I have a colorblind male. So X little ny. So this X would be little n, and my boys are taken care of. So what are the chances of having a colorblind daughter? 0%. What are the chances of having a colorblind son? 0%. What are the chances of having a normal color vision daughter? 100%. Whoops. 100%. What are the chances of having a normal color vision son? 100%. Now my daughters could be a carrier, but they're still normal. So a colorblind male that marries a homozygous normal mother or female, if they have kids, there is no chance their kids can be colorblind. Because again, it is linked to the mother's side. And since the mother is homozygous dominant, it's not possible to have an offspring with that disease. So let's do one more and then you'll do it yourself. Last one. I know this is getting long. We're going on 30 minutes here. Okay, number 2. And of course, if this makes sense, you can kind of work ahead. But a carrier female, so let's think about that. So it's a female, XX. She's a carrier. She's still normal, so she's X big N. I'm going to erase this here so we can see it just a little bit better. All right, a big N, little n. Normal, but a carrier, all right? Marries a colorblind male. So, whoops, that's a female. Let's not do that. Okay, there's my male. Let's make him colorblind. So I go over to my Punnett square. X. Doesn't matter who goes on top and on the side down. There's my Punnett square. Got all, everything. Now it's a matter of where do the ends go. All right, the female is X big N little n. The male is X little n y. So in this box, it's going to be an N and little n. All right, that goes there. That goes there. Okay. The second box, X little n, X little n. This third box, X big N y and X little n y. So the question are, what are the chances of having a colorblind daughter? Now there's two ways to look at that. You can look at this out of the whole, out of the four, or you can look at this question as if there is a daughter, there is a 50% chance of having a colorblind daughter. The second one, what are the chances of having a colorblind son? So if we're looking at just the child in general, then of course it would be 25%. But if we're just saying if they have a son, what is the chance that they will have a colorblind son? And that'll be 50% as well. And that's the case for the rest of the two. Okay? So there's two ways you can think of it. If you have any questions when you're doing these, just go ahead and ask, and I will give you the opportunity, or I'll share the information with you of what I'm kind of looking for. Okay? Since there are two ways of looking at that. So go ahead and finish the rest of these problems. If you need any help, ask, but go ahead and answer all of them, all the way to number 12 in the back. And please do this all by the end of class. If you do not finish, then you need to finish these for homework.